Start the music. From Microbe TV, this is TWiEVO, This Week in Evolution, episode 102, recorded on May 1st, 2024. I think it's like Labor Day, International Labor Day.
Oh, yeah, international. Coming up on Memorial Day, I guess, by the end of the month. But greetings from LD Lab Studios.
Hello, Nels. Welcome to TWiEVO Live. I'm Vincent Racaniello, and joining me from Salt Lake City, Nels LD. Welcome, Nels.
Thank you. Jumped the gun there.
No worries. We
from
can do
LD
anything. It's our stream. We can do whatever we want, right?
Yeah, and we're into triple digits, 102. So I feel like we've got a little bit of flexibility to operate by our own devices here.
Welcome, everybody, to your afternoon Evolution live stream. And let's welcome our moderators today. We have Barb Mack, who's in the UK, and we have Tom, who is in, Western Oregon today.
Welcome.
We have Andrew, who is in New Zealand. And that'll do it for today. Thank you for moderating with us. We appreciate it.
Really appreciate it. Afternoon on the Eastern Seaboard, but different times of the day all over the world. Great to be together, everyone.
I need to put your name there. Where is it? There we go, Nels.
Looking good.
At least I noticed it. Yeah, it's like in the mid 60s, high 60s today, and a little bit of sun. It's nice weather. It's very nice.
Similar here.
Let's
go around the horn. Yeah.
Yeah, let's tell us where you're from, folks. And they're not a huge number of you, 30 or so, but tell us where you're from.
Yeah.
We love to hear where everyone is coming from. Claire is from the UK also. So now it's like, what is it? It's 7 p.m. Not too bad in the
evening,
in the
yes.
UK, right? Let's see. Joseph is in Southern Ontario.
Great. We're up to 4 countries at least.
Kang is in Chicago. Here's another country. M.E. Wheeler from Helsinki. Yeah. I might be in Helsinki in June, M.E. Hopefully I'll get there, and I got a weekend to kill in Helsinki. If you want to, what is the word? Connect?
Connect.
Yeah, you can show me something to do in Helsinki. I'll be on my own there. So Helsinki, you can contact me offline, vincent@microbe.tv. Or not, you don't have to, because you might be scared of me. You might be thinking, this virology guy, why would I want to talk to him? So no worries.
I can vouch here. It's good to be in vivo with you, Vincent. It's always a positive encounter. Lovely sunny evening there in Helsinki. 13 degrees on the centigrade scale.
Jeremy is in Seattle.
Welcome. Some regulars. Our core audience has arrived.
Angelica Di Nova Lipa. That's a great name.
Yeah.
Manhattan. Welcome. Probably not far from the Incubator, which is on 7th Avenue and 30th Street. Ruby. Yeah, I guess Ruby is from Albany, New York. And Christopher Robinson. Isn't that a Winnie the Pooh character?
Indeed.
From Scotland.
Great.
Surah is in Abu Dhabi.
Welcome.
That's pretty far away. It's 6 p.m. in the UK. That's right, because we do that dumb spring forward, full back with the clocks in the US. Nobody else seems to do it.
That's right. It's a small world. The work that we're going to lift up on today's TWiEVO 102 happened in Abu Dhabi. And so we've got a local connection to the work that we'll be talking about just in your neighborhood.
Rob is from Calgary. Cool. It's way out west and north.
Mm-hmm.
MK is
Welcome.
in Greece. Oh, you're on vacation because I know you're normally in like uh, Eastern Massachusetts, something like that.
Mm-hmm.
Yeah, and
That's great.
it's 8 p.m. there. I'll be asleep for office hours. Yeah, I probably will be too. Not just kidding. I will be wide awake. I get going when I do office hours. Famida
Yeah.
is from Chapel Hill, North Carolina. Very, very cool. Oh, Barb Mac says they do the time thing also in the
Hmm.
UK. So I
Okay.
guess some countries do and some countries don't.
Mm-hmm.
Here's Les, one of our mods.
Great.
Thanks for joining us, Les.
Yeah. Good to see
Um,
you.
and while your folks are at it, please hit the like button. That will help potentially attract some other people here. You know, we'll
Yep.
feed the algorithmic beast.
That's right. There's no, and in the algorithmic beast, I don't think accounts for a trend line here, which is spectacular. I think I counted 8 to 10 different countries from around the world
Nice.
with our small audience. That's pretty spectacular, I have to say. We're
Yeah, I
coming
think it's
from it all
great.
over the map.
I
Yeah,
love that people are coming from
me
everywhere.
too.
Totally love that. Yeah.
Yep.
Yep. All right. So today we're going to talk about kelp, right, Nels?
Yeah, kelp and its cousins, so to speak here. So we had a paper recommendation based on a provocative press release. So Kathy Spindler, this week in Virology's iconic figure, um, got this onto our radar. I don't think, Vincent, we've considered any papers in the journal called Molecular Plant, and so this is we're charting new territory here, and a pretty cool comparative genomics approach that we'll dig into together as a group. Microalgal deep genomics illuminate multiple paths to aquatic photosynthetic multicellularity. So a few buzzwords there that we'll cut through in a little bit. But to begin, let's talk about who did the work. So David Nelson, Alexandra Mystikou, a cast of characters in the middle, and then senior author Kurash Sali Ashitani. And I wasn't bluffing. So the first and last authors here, the Division of Science and Math, NYU, New York University, but on the Abu Dhabi campus in UAE, United Arab Emirates. And with some assists here from some names that show up in the evolution of multicellularity. So William Ratcliffe is one. He's at Georgia Tech in Georgia here in the U.S. And people from all over the world, as you can see, sort of here in the byline. So the same way the TWiEVO audience reflects people all over the world, so does science in general, including this example, these great collaborators collaborations that come together with people with expertise in different fields. Really, a great crew to do the work. And Mark Martin, a podcasting aficionado, newly minted podcast, which is great, worth checking out. Microbial Matters. William Ratcliffe is here. Someone who's done some really interesting work in multicellularity in fungal genomes or fungal species, and pushed it into some provocative experimental space that's generated a lot of conversation over the years. So we've talked a little bit in the past, Vincent, about multicellularity, the evolution of multicellularity, mainly focusing on a totally different critter, choanoflagellates. And so
Right.
Nicole King, right, is a champion here who has really built this genetic model system or experimental system from the ground up, including its genome a long time ago, a lot of experimental tools, and some real interesting surprises, collaborations between other microbes that choanoflagellates encounter and feed on, and how the signaling, the cues come together to trigger real-time transitions from unicellularity, or the status of being a single cell throughout your replication cycle, all the way into multicellular sort of conglomerations and the ability to develop that sort of right before your eyes. So, sort of this reversibility of multicellularity in some cases, which will come up for the creatures that we'll be considering or focusing on TWiEVO today. But also some really different sort of, genetic backgrounds and evolutionary paths. And it's really, I think, in that power of the kind of comparing these, where you can start to see the independent emergence of multicellularity starts to fill in kind of the rule book of the flexibility or the ability to do this. And I think, just from sort of a philosophical standpoint, what's the advantage of multicellularity versus being unicellular? And you and I,
you get to have a mouth so you can eat.
I was just going to say, yeah. There's not a lot of single cell critters out there that are podcasting, for example. So we're enjoying some of the fruits of multicellularity in this conversation that we're having together today. So here the focus is going to move into algal species, and already, just beautiful, diverse species. So the three main categories here, this is at the level of phylums. So, let's think back to our sort of taxonomy from biology class. Did you have that, I guess, the mnemonic device, is that right? So kings play chess on funny green squares. Does that ring a bell?
No.
Vincent?
Kings play chess, KPC, G, S. What is it?
Kingdom, phylum.
Oh, I just memorized it.
Class, order.
Kingdom, phylum, class, order, species, genus. Yeah, variety.
Genus, species.
Genus,
play
species.
chess on funny green squares. So that's how I remember it, kind of drilled in back in middle school or something like that. And so we're on the P, the play. So phyla, there's 3 phylums just below the kingdom level of algae. And the fact that it's so far up the taxonomic scale there speaks to the diversity of life forms that fall into this category of algae. So a lot of aquatic critters, some with a resemblance to plants. And so, the authors point out the great diversity, not only of sort of species, but also just sort of lifestyles. And so you've got algae like kelp that can be 65 meters long, all the way down to microalgae that are single cells that are, basically millimeters or even, approaching microscopic. And so that's sort of a first, sort of classification I think is important to get on the table here, is the difference between simple and complex multicellularity. And so, and some algae, sort of all of the above for algae. And so in that sense, I think it's really makes sense to point the kind of genomic tools towards all of this diversity, not just for multicellularity, by the way. Yeah, go ahead.
I just wanted to point out that we have, Tom says, kings play cards or fight green serpents.
Oh, great. Yeah, another variation on this mnemonic.
Or Sura has, dear king, Philip came over for good soup.
I love it.
It depends which one you remember, right? I didn't memorize any of them. I just memorized the thing because...
Well, that's doable too, right? Yeah.
When it's not too complicated. Kingdom, phylum, class, order, genus, species, variety. But most people don't do variety, so just stop at species, right?
Yeah, but I think you missed family. So there's
kingdom, wait, kingdom. Kingdom, family. No, kingdom. Phylum, family.
Nope. That's where the funny green square is. It puts it right before genus.
Phylum. Oh, kingdom, kings, kingdom, phylum, class.
Order, class.
Order. Phylum. We got already phylum. Order, genus, species. I guess I should have used one of these mnemonics. Yes.
There you go. I rest my case. I rest my case.
I used to just rip it off, and when I was a student, kingdom, phylum, class, order, kingdom, phylum, class, order, genus, species. Is that it? Kingdom, phylum, class, order.
Family slipped off the radar again. So my middle school science teacher is looking better and better by the podcasting minute, I have to say.
I was so proud that I didn't have to do that.
That's right.
Just use, Mark says, just use virus names. Yes, I should figure out some virus names. Yeah,
exactly. Good call. So getting back to the multicellularity. So here complex is kind of generally associated with the ability to conduct photosynthesis among the algal species. And so so that's one of the first definitions, and they're suggesting that there's sort of a key transition here to complex multicellularity that's going to involve a lot of genetic changes compared to some of the single cell relatives. And so that's sort of, the kind of a hypothesis, I think, that was sort of guiding or helping motivate the focus on multicellularity. I think you can just, like, to begin with, to just make the case that getting good genome sequences, annotating them and analyzing them to some degree for this, major set of life forms, phyla of life forms that are sort of undersubscribed in genomic knowledge, like that alone, like, count me in already. And so we'll talk about that. We'll kind of run through that. And then, so why did Kathy Spindler, celebrity podcaster from TWiEVO, put this on our radar? There's a connection to viruses. Getting Mark, to your point there, not only do you could use this as a mnemonic device, but the viruses are sequences, genes from viruses are showing up in abundance in a lot of these algal genomes. And so they really, I think they kind of start through that lens of multicellularity and really try to fit in the virus sequences here to how it might tell the story about those transitions to single cell to multicell, how that played out independently in the three phyla, the red algae, the brown algae, the green algae. And so we'll unpack that a little bit here today. We'd love to get the feedback, questions, responses from our live stream crew here, our community, and then see where we come out here in terms of, do they deliver on some of the connections between the abundance of the virus sequences that are showing up across the board in these diverse genomes, and linking that sort of in real ways to the process of becoming multicellular, evolving multicellularity.
So Tom says this is not a quick read.
Yeah, I agree. And that's it. It's an interesting point, Tom, because I think, the paper does a good job of sort of comprehensively doing comparisons and going through, but why is it not a quick read? It's a lot of details in there. I think for, like, from the casual reader to the sort of genomic aficionado, is there really a storyline? I mean, so does it tell a story? And I think when you sequence 100 genomes, it's pretty hard to tell a story. I mean, you're swimming in data. And so, of course, they're using multicellularity as sort of a touch point here, but are they convincing in that? And so, already I think we're kind of on the same page in a sense of how do we grapple with a study like this? The science author, Carl Zimmer, used to speak about sort of the era that maybe emerged 15 years ago as the technology really became somewhat democratized, the ability to sequence entire genomes and then analyze them. So there are all kinds of papers, sort of breathless titles about, you've sequenced the genome of a bird and it unlocks the secret to flying at 50,000 feet and so in the whole field of genomics, there's this, I think, impulse to try to tell a story, but you're sort of faced with this sort of mountain of information. And the challenge here is, how do you squeeze meaning out of that? And so here, I think evolution. So what's a reason that when Kathy suggested this to us, that we jumped on it? In part, it was, in the hopes that kind of evolutionary perspective can really help to sort of unlock some of that meaning, to kind of go into the into the story of what some of these genes mean. I think delivering on that is easier said than done. And so that's sort of the work of TWiEVO 102, is sort of walk through that and sort of gauge what have we learned here? What's the information here? And let me just stipulate out front. So, it used to be kind of the era of you'd sequence one genome, report it in a big kind of flurry of a press release, et cetera. The bar has kind of risen just as genomic technology has become even more accessible and even more powerful. And so here they show up to the table with 110 genomes. And so, really kind of a tour de force in that sense of being able to gain that kind of information, even for a single paper, to really kind of cast light on what's hanging out in the genomes of all of these 100 or so different species of algae from the three phyla, representing, billions of years of divergence. So, yeah, and times have changed since 1971. I came on the scene since then, for example, but it's really true. And so we're kind of glimpsing at the possibilities here of this technology to really understand algae in a whole different way based on sort of the recipe book of what's in these genomes. Okay.
So before you jump in, Vanity
Yeah.
Nutrition has joined us. Thank you for
Great. Another
coming.
moderator here. Appreciate it. Yeah.
Mark Martin says it's not a family tree, it's a family cloud due to horizontal gene transfer.
Yeah, it's a really good point. And and that's the fact that all of these sequences that are appearing from viruses are sort of infiltrating these genomes. That kind of throws sand in the gears of simple family tree like relationships. Basically, you start, if you do kind of like a tree of life. Now imagine all these little dotted lines that are between this lineage and that as each gene jumps over there. If you're just building a tree off of a single gene, but it's jumped between genomes, that can really kind of fool you into thinking that things that are actually less related to each other might appear so if that was a recent horizontal gene transfer event. And so we've talked about horizontal gene transfer before. So this breaks Rob, we're on actually wait till the title drops on this episode. We're on the same wavelength here. So we've talked about vertical transmission, basically genes moving from parents to offspring. Horizontal transfer is when you sort of break that rule book and somehow genes are getting between species without reproduction happening. Horizontal transfer can actually happen during reproduction as well, but separate from that sort of main conduit of gametes coming together and genetic information moving forward. So here they start with, and so it's kind of just 124 genomes. So started at 14, now there's 124 on the record because of this study. And the punchline is that some of these genes that they propose that are involved in multicellularity or might have been key to some of those evolutionary transitions actually originated in viruses and somehow showed up in these genomes, kind of the raw material of natural selection to act. So there's each horizontal transfer event might be considered as like a mutation, but it's like the wholesale acquisition of genes or multiple ones, or domains of genes. We'll kind of talk about that. The notion that these gene sort of regions or domains are coming together in novel ways in the algal genomes. They have some signals of that happening pretty prolifically. And so the punchline that they get to is that some of these genes involved in multicellularity originated from viruses and then were brought in. This is kind of an emerging theme in evolution, is that viruses are probably really good vectors of horizontal transfer. At the same time, I maybe throw a little bit cold water. So, on the notion of horizontal transfer, there have been some false starts in the field. So maybe one famous example was when the tardigrade genome was done. This is the so-called water bear that you can launch into outer space. It lives like without any oxygen and you can kind of recover it, all this kind of stuff. There's a pretty provocative note when that genome was sequenced that there might be as many as like 10% or 5 or 10% of the genes might have been horizontally transferred. Turns out in the sequencing was fine, but the analysis was a little bit lacking there. And so it was sort of debunked, and a lot of the genes that were proposed to be horizontal transfer events were actually either contaminating or sort of it was fooling the computer algorithms. That is really a big part of this. And so the record's corrected, and this is like sort of a useful for the field, a useful sort of learning moment to really think carefully about the sort of the chain of custody of the genes that you're identifying. Where are they coming from in these samples? So a couple of things that these authors do to sort of get out in front of this. First, the 105 species that they tackle are all available from culture collections, right? So you can kind of go on the internet, go to these culture collections, I think from several places around the world, and order algal species right to your laboratory. I mean, I guess you can DIY this, probably order them to your basement, and sort of, take these on as new pets or, critters so this is really useful, right? Because a lot of these species have been taken from the wild environment, which could be messy, complex, sort of chaotic, have been sort of semi-domesticated, and then are available, and they're maintained, right? And so the things that you sequence are now available to other people. And so that's really important because it gives you I think it gives the data set ultimately, which is massive, a level of quality where you can go back. If you see something interesting as a scientist, as an evolutionist that catches your eye, you can go back to that, say in order that same strain that's being propagated and really know that you have kind of the same thing that the authors here, that the scientists here have been analyzing. So that helps. And then, but the second kind of technical piece here that shows up probably more in the methods than kind of in the main text here is this pipeline for what they call decontaminating the genomes. So I think I hadn't heard the phrase decontaminating as a as sort of an annotation step, but I think I was like working backwards to their acronym for their pipeline, which they call BLEACH. And so the B is for BLAST, which is a blast search. This is basically taking gene sequences, comparing them to things that we've seen before that people have deposited on public databases. And then they add some more steps to that to try to like get out front again and remove genes that are sort of are contaminating, in a sense, those samples that they got from all of those culture collections. And so, in this case, they point out, and this is again in the methods section of the paper, that they really took a strong hand to remove genes from bacteria, from fungi, and from archaeal sequences. And so the notion would be these could be like hitchhikers on the kelp, or hitchhikers on the different species of algae. And so the danger here, or the challenge, I should say, maybe not danger, the challenge here is, as when you're sequencing a genome, you're also sequencing everything else that kind of comes along with that. And so you're trying to really enrich for the genes that are in the genome of the target species, the algal species, the 100 plus that they're looking at here. And yet, even in these sort of domesticated samples, there'll be all this other stuff. What they didn't mention in the methods, and it was a little thin in some spots, it's long, it's because there's a lot of info, but maybe some key info, is what do they do about virus sequences that show up, right? Because this ends up being sort of the main thrust of the paper in the end, is that there's all of this horizontal transfer of virus genes into these genomes. And I think, don't get me wrong, I think they have some good evidence for that. But is every case that? Are there potentially exogenous viruses that are infecting some of these algae? Probably yes. And given how some of the analysis pipelines, the annotation, so when you go from all of that raw sequence to try to understand, okay, this part of the genome, all those A, T, C's, and G's encodes a gene, and we think it's from a virus, is that because it's sort of co-housed in the cells that were being sequenced, or is it actually integrated into that genome? There's no sort of separate genome from a virus.
You could make an algorithm to sort that out, right? So you could say the viral sequences have to be next to a cellular sequence in the same read, right?
Yes. And here's where, from my reading, at least, of the methods, I think they do all Illumina sequencing, which is short read sequencing. So it's harder to get that information, actually. And as you build the libraries, actually, you can be fooled. So even if you take some of this stuff into account, just as, so you're part of the kind of the technical behind the scenes work here is that you're getting all of the DNA, you're isolating the DNA from that species, and then you're doing several enzymatic steps to basically prepare it for the sequencer to be able to step through and give you back for each fragment of the genome. You're also sort of short read, you're cutting it into small fragments, and you're only sequencing, say, 100 or a couple hundred base pairs at a time. Sometimes it's bigger chunks, but you're just sequencing the ends of it for a couple hundred. You've got that connection between it, and you can start to overlay this. And in fact, they get to 100x coverage for these 100 plus genomes. I mean, this is an incredible amount of information there. But again, that's like, it kind of even raises the stakes of the challenge. So you might even, if you built that algorithm that you're suggesting, Vincent, which in some of these exist, even then you can be fooled in some cases if the enzymes are just splicing together some of the viral sequences to the sort of, right. And that's part of what was in play with the tardigrade story as well. So they talk about removing bacterial, fungal, and archaeal sequences. And fair enough. Although there could be some horizontal transferred ones maybe that are, it's also like, if you're bleaching genomes, as the pipeline is called, is there sort of a side effect that you're taking some stuff out, or you're including some stuff that you would actually want to take out? I think especially as you're doing kind of this massive survey, it's a little bit hard to really maybe nail down the sort of exact level of getting it just right. And certainly there'll be a lot of revisions if people pick up, if you're, someone is in some of the species they chose are really model systems for things like brown algae, where there's a lot of experimental sort of efforts to understand the biology here. And so as someone does a deeper dive there might be, some real strong motivation to do increasingly sophisticated annotations of those genomes, or to describe what's there versus what's artifact from the genomes, what shouldn't be there. Okay, but they do, like, the power here, I think, is at that sort of 10,000 foot view of having all of these genomes. Now you're able to start to compare these things and to ask, what are the protein families that show up? And does that start to, how's that comparative between different species, different lifestyles, the emergence of multicellularity, as they try to kind of date that and correlate that to the types of genes that show up and might be involved in the ability of a single cell critter to gain multicellularity. And so, they describe then, as they're sort of correlating those lifestyles or life histories to what's sort of encoded in these genomes, they propose that there are some, like, unique genes that might be involved in that transition. Bit of a leap here, but they identify something like 1,000 unique gene families, meaning that you find that, like, in the brown algae that are multicellular versus non-multicellular, different from the ones in the red algae, multicellular versus non-multicellular, same for the green. And at the same time, they sort of lift up 9,000 shared protein families. That's a lot between these that they propose might have some role, maybe directly or indirectly, in the ability to be or to develop as a multicellular organism.
It's over 9,000.
Yeah, it's a lot, and it's.
No, I don't know if you get that reference, Nels.
Oh, no, I don't actually. Where were you going with that?
Well, it's an internet meme.
I see.
Which is from 2005. It's quite old. It's some episode of the Dragon Ball Z anime, where one guy says to what does the machine say about this, and the answer is it's over 9,000. So it's actually over 8,000, but people have made it over 9,000. So whenever someone reads, Barb Mack got it. Whenever someone says 9,000. We even have a podcast episode entitled that.
Nice.
As I can't resist saying it's over 9,000.
I gotcha. Yeah. But again, sort of squeezing meaning from this, like, we're still flying at a pretty high altitude in terms of just sort of, but I think, in this early analysis, they're really just trying to get the playing field of possibilities. They then go on to get a little more, to sort of focus down or get a little closer to the ground level here. So they go ahead and start to do, and this is, what I think, anyone doing genomic analysis would do, is to start looking at gene ontology analysis. So to try to connect some of the gene families to what they actually do, the categories of functions that they have. And so, that actually immediately pops out entire classes of genes that are known to be involved in the development of multicellularity or the regulation of multicellularity. And they sort of say, you find some of these, the usual suspects, so to speak, in these algae genomes, all three phyla. Red,
by the way, Nels,
green, red, blue, and not blue, brown. Yeah. Sorry, go ahead.
Let me show the map here of where they got these samples, because it's really amazing. It's just global, right?
Yeah, good idea. And this also, is an important point that a lot of algal species, a lot of algae you find in, of course, aquatic environments, including in the ocean around the world, and not just sort of in temperate places. Look at those dots, on the tip of South America there, into the Antarctic. Lots of cold adapted species all the way into warmer waters. I'm looking in the Caribbean there and kind of wishing I was on the beach in San Juan, Puerto Rico, or something like that. We have some algae from that neck of the woods. Right below that, Vincent, I think, is a really cool diagram here, which gets at the different habitats for the 110 that connects that to the temperatures and the climates from tropical to polar. Tries to relate both the environments and the phylums. It's really gives you a sense, the habitats. This is great, right? There's some that were originally cultured from oysters, for example, or from aquariums, in some cases, rivers, salt marshes. So all the way from fresh water to seawater with brackish water in between. And this really kind of points to just the diversity of life again. And the fact that we're, from a genomics standpoint, kind of up until this study, we're really flying blind with only 14 genomes known, given the diversity of environments, adaptations, all of the above. And so, I mean, what an amazing resource to have 100x coverage now on more than 100 genomes sort of sampled across all these places and representing some of the diversity of algal life forms out there. So, that's, I think, this also illustrates, so now as they're kind of getting at those functions or finding kind of the usual suspects, the genes that you'd expect that have been pointed out as being part of multicellularity, the development of multicellularity in other species. As you do these comparisons, you're sort of, not sort of semi-limited to sort of existing knowledge as you try to sort of put these genes into categories where it's known. I'm certain that kind of hiding in these gene families, and this is where the authors will move as we kind of continue to step through it here, are sort of new ways of becoming multicellular, or genes or functions involved in that process of living your whole sort of reproductive life as a single cell into becoming multicellular. And so, some of the not usual suspects that they're trying to kind of enrich for those classes are kind of pointing to new biology and ways for this to happen. So in particular, they kind of focus in on brown algae for a couple of reasons. So one is they infer, and they do a lot of molecular clock analysis that we won't go too deeply into, but the brown algae actually were pretty recent. There are newcomers to this transition of being multicellular, that this happened, in something like the last couple 100,000 years, which from an evolutionary standpoint is sort of like a blink of an eye, whereas some of the red algae species, the green algae, the notion there is that multicellularity has been around for more than 1 billion years, some of the early days of sort of the diversification, the descent of algae. And here's where, having some model systems, so this genus Ectocarpus, and I don't have on the tip of my tongue the actual species that has become the model system here. This is a brown algae that's been used for a lot of experimental work in this entire class, in this entire phylum, I should say, of algal species. And they already are noting lots of virus-derived gene insertions. And the fact that this is that are unique to the brown algae here, the fact that this happened more recently, they're sort of, kind of almost putting a highlighter pen to underline, like, this is a place to look for maybe some new clues about this recent acquisition of multicellularity for this entire phylum. They also, here's where they're noting, so kind of that we've talked a little bit about simple multicellularity versus complex as it relates to algae and the photosynthesis that they're doing. They also note that there's like these reverse transitions, like some multicellular algae will go back to unicellular. It's sort of this evolutionary two-way street. It's an important point for, like, as you're trying to link some of the functions together, if this is a regulatable process, how does that work? And so I think throughout the paper, they raise these like really important points. I think that where they maybe fall a little short is connecting that sort of to the gene composition, the comparative to the actual functions. And, I'm not trying to do sort of vicious podcast peer review here. But because let's remember that getting 100 sort of new genomes that really are like kind of carefully curated across all of this diverse life is pretty impressive just as is. And I think the real promise is in building off of the sort of sightings that they have and the data that they've collected to do even more incisive comparisons into the future. So I thought I would just do maybe a quick, just sort of point by point of some of the highlights that they really, sort of are getting at with the analysis.
This figure 2 would be cool to show and chat a bit about. It's an interesting way of showing their data. You want to do that?
Yeah, good idea. So this is right. And so this is a nice way, I think, to graphically illustrate sort of the power of the comparisons here. So they're showing it as what they're calling ternary analysis in this sort of triangle form. And so the points of the triangle are formed by the green algae on the top, the red algae to the lower right, and the brown algae to the lower left. And so this is basically, thinking about gene composition. That's what's shown in that sort of purple graph, that heat map, the total number of gene families and where you find them relative to each of the three phyla that are represented at the points of the triangle. And so, yeah, and then this is kind of putting some names to the faces, basically. What are the PFAMs, or the protein families, that are showing up that are enriched? The size of those purple circles indicate sort of the size of these families, and then they point out, some of the functions, or the protein domains that will have functions. Some of these I think are more informative than others. So ankyrin, for example, are repeat proteins that are sort of notoriously known for promoting protein-protein interactions, which of course is sort of a fundamental part of biology, but can really be, underlying a lot of biological outcomes, not just multicellularity, but just sort of how proteins work together in a cytoplasm of a cell. Here we're starting to see some hints of like the magnitude of all of the potential viral contributions. So one of the big triangles there that is sort of in the northeast quadrant, I guess, between sort of shading towards the green, but also, some representation in the red, are the reverse transcriptase genes.
Yeah.
So this is that virus connection, right, Vincent? So the reverse transcriptase, these are super famous genes among virologists for their enzymatic ability to take RNA and reverse transcribe it back into DNA. Again, speaking to some potential mechanism here. How do virus genes ultimately get in to host genomes? Reverse transcriptase or integrase genes which is right below that, give you sort of the motion, in a sense, the biological motion or the genetic motion to actually execute on the acquisition or the capture of genes from virus genomes to host genomes, or even between host to host in some cases, if the viruses are acting sort of as intermediaries in that process.
So Nels, are these genes here because they're in the multicellular organisms, but not in the unicellular?
Yeah, correct. So that's what they're enriching for here.
Okay.
Yep. And so, lineage, or the way they describe it, is lineage-specific protein families in the clades that form the basis for independent paths to multicellularity. So they're doing, it's a lot of information but they're trying to enrich here in genes that they see in the multicellular for all three comparisons versus the unicell, the microalgae.
So Don wants to know what the size of the circles means.
Yeah, so the size is basically the I think actually the colors help with this too. So the darker the color of blue, the bigger the family, the number, just in quantity of the genes that they find. And so that's where they, why do they underline sort of the virus contributions? Because reverse transcriptases, some of the RNases that, so these are enzymes that are processing or chewing up RNA. A lot of these have their direct connections to viruses or retrotransposons or endogenous viruses, sort of an intermediary between the relationship there. The major, so ankyrin, as I pointed out, so that's another where the balls are big and the numbers are in the thousands based on the heat map. That's probably non-viral, but the fact that you have those big purple circles that are involved with virus, that's kind of why they draw that connection here.
So Nels, how do we know that these drove multicellularity or happened after multicellularity? like the retroviral genes. I'm not aware. You would know this. Are there retroviruses that infect unicellular organisms?
Yeah, the implication is yes. What those look like, I think that's a little more cloudy. But I think your question is exactly the right one, which is where sort of what's the story here? It's sort of these comparisons and these enrichments of gene families, but actually tying that to the process or when that was happening. So, for example, like if we take the reverse transcriptase, like I think for me at least, my first impression is that kind of gives you the ability to move other stuff around to sort of enhance horizontal gene transfer. But whether that relates to multicellularity, I mean, so the way they're directly tying that, right, is to say that we see these things in the multicellular ones, we don't, or they're right in the unicellular. So that gives them a leg to stand on for sure. But, there's all kinds of, I think, additional explanations, including ones that the authors, they're not trying to hide this at all. But how much is this involved in maybe, because it's correlation? Yes, you see it enriched in multicellularity, but does it speak to sort of the immune responses and the evolution there, which is,
Yeah, sure.
a massive category that makes a lot of sense for thinking about how natural selection can act in really sort of drastic or dramatic ways over short time periods. And that's, basically, are you contending with the infectious microbes that could really impact your fitness in a negative way? There's also, another massive class of functions where natural selection is acting is in the ability to reproduce. And so, if you have more kids, those are the genes that will sort of be enriched in populations as you move forward in time. And so here, that actually, again, ties directly to multicellularity, because by definition, right, if you're a unicell, your genomes are basically, the somatic genome and the germline genome are together. There are some exceptions. Some of my favorite critters, the ciliates, that actually have two nuclei, a macronucleus and a micronucleus, to divide the somatic genes, the ones that are sort of like the daily jobs of a cell, from the reproductive genes or the germline genome that goes on to the next generation. This is what, this is sort of one of the massive advantages of multicellularity, is that you, in different cells, become germ cells from soma. So like our skin cells, right, are not going to reproduce. These are so-called somatic cells. Whereas the eggs, the sperm, those are the germ cells, and the genomes, it's the same genome, but it's been sort of separated into specialized cells that then, have those jobs. And that's, again, probably folds into immunity in some ways. But that also is going to influence fitness here. So those, for me at least, those sort of major categories of reproduction, which obviously necessarily involves or is entangled with multicellularity and some of the specialization of germ cells, to sort of immune functions, immune responses, how that all plays out, or what's really driving this, I think is still a bit cloudy. It's complicated, it's complex. And so they do a lot of, careful comparisons to try to point out the impact of multicellularity. But I think it's good to keep an open mind as the work advances here.
It's hard to support this experimentally too. You can't take a unicellular organism and try and convert it, right?
Yeah. So that's what, that's what's really compelling and interesting about the Choanoflagellates, Nicole King's work, and others that have come from her group, and some other labs thinking about that. And also the Radcliffe lab, that's one of the authors here, and some folks in his group, where they've, taken fungal unicells, done experimental evolution to select for multicellularity in some cases. And there, the big question marks become, is this true multicellularity? I think, it really raises a lot of interesting questions. Have you modeled that process in the lab sort of in a way that happens in nature? And even if the answer is not necessarily 100% yes, are some of the, so in my lab, we do a lot of experimental evolution with viruses and host cells and other infectious microbes. And we're actually, we're conceding that we're not capturing the complexity of real life. But the question becomes, are we setting up the experiments to find things that actually kind of give us sort of a sense of what's possible, or the rule book about how some of these outcomes might happen? And so, yeah, there's definitely the possibility to do that. I think you could imagine, actually, I don't know, that's where I think it's really important how they, where they're pointing out those transitions back from multicellularity to unicellularity. Those are probably the right species to think about kind of taking that experimental approach that you're describing. Yeah. So, a couple of kind of the highlight reel here of the enrichment of those virus-derived genes from the multicellular species that they've sampled. So the green algae have probably some tie, just based on the relationships. They're a little bit closer to land plants, green plants, some of the photosynthesis there. And so they kind of highlight maybe some of the shared gene families that could also be enriched or involved in multicellularity. For the red algae, a lot of virus-derived genes. That's where those reverse transcriptases, those RNases, are showing up. But the functions, as you're pointing out, not so clear. They actually did some comparisons with fungal genes that have been implicated in multicellularity. So there's like some of very similar characters of filamentous fungi, where cells start to divide in ways where they don't kind of finish the division. They divide and continue to divide as a conglomeration or a cluster. And again, they see sort of shared genes. In this case, they point out from some parvovirus genomes. I don't know about you, Vincent, but as a virologist, when I think of parvoviruses, I think of like dogs and cats, where the certain news headlines have been Colin Parrish at Cornell is sort of famous for bringing the parvoviruses to the forefront in his career. And so, but parvoviruses are prolific and are, kind of infecting not just mammals, but it appears also algae and perhaps contributing, given that sort of echo of some of the genes that have been implicated in multicellularity for fungi. That could be part of the story here as well. The other theme that they kind of highlight is, gene gains and losses. So the retention rates of gene families is something that they calculate. Again, trying to compare the microalgae, the unicells, to the macroalgae, the ones that get bigger because they're filamentous or have done other sort of tricks to become multicellular. And they see a correlation with gene gain, that they're connecting to like when those events happened, right? So the ancient events that you see in the modern examples of red and green algae, and tracing that or kind of contrasting that in this case to the brown algae. They also, again, kind of narrowing in on the gene functions that would make sense for multicellularity. Cells need to hang together. They need to adhere to each other. And so some of the genes that are involved in making, proteins that can encode, or their function is adhesion. So things like adhesosomes, or genes like integrins or cadherins that are sort of famous in mammalian developmental biology for allowing multicellular cells through the process of development to hang together. Also important, obviously, or maybe not so obviously, but as you think about it, for multicellularity or transport proteins that are sitting at the membrane. So as you become more and more multicellular, one of the challenges that arises is if you're a cell in the middle, how are you getting nutrition versus a cell kind of on the exterior that's exposed directly to the environment? And so, as you kind of go down that road, as our species has, we have things like circulatory systems and all of this sort of complex specialization. For some of these algae samples or examples, that are still in aquatic environments, but might now, as they become more multicellular, are they transporting amino acids? Are they transporting other nutrients in ways that would make sense for sort of more recent, in the case of the brown algae, in those more recent cases of transitioning to multicellularity? And here, the metabolic pathways are important as well. And they do a little bit of a dive in there. The trick here...
Basically, you have to consider the biology together with the genome information. You can't use it in its own standing, right?
Yeah, agree. I mean, the trick here, I think, is also to sort of at what level are you thinking about the evolution of multicellularity? So, for example, some of the integrins and cadherins that are involved in adhesion are probably, super important for like the life histories or the sort of style of these algae. So if a lot of which like cling to rocks, right, and might be sort of constantly with waves sort of constantly crashing down on them. And so, you've got the environmental sort of selection here. If you've got a really good specialized cell that can cling to a rock, these are some of the genes that are going to be encoded to actually conduct that function. And so
a fish can come and eat it. That's good.
Right. You're setting up the buffet line. And so you've got this, like all of these like complex environmental factors. And so, what's sort of the proximal cause for the development or regulation of multicellularity, and what's sort of the output? Like if you have these specialized cells that are expressing some of these cadherins, et cetera, that are involved in, clinging to a rock, is that really multicellularity? It sort of depends on your definition or what level you're kind of thinking about it. Okay. So that's sort of, I think, the kind of the grand landscape that they sort of lay out there. And then finally they return to these virus-like genes and kind of drill down a little bit to talk about some of the functions here. And the notion that the horizontal transfer of the virus families are sort of facilitating the genetic diversification, which is, I think, is clear and sort of supported by a lot of other work out there. But again, sort of raising that question, are there ones that are enriched or enhanced for that transition, that evolutionary, leap in a sense to multicellularity? So how do they do this? They use another form of, sequence comparisons, in this case, hitting databases that are sort of specifically curated for viral genes. And so instead of PFAMs, the protein family annotations, they're looking at VFAMs, the virus families of proteins, and doing these sort of really sensitive sequence comparisons, because one of the challenges here is that from a primary sequence level, this is all the letters, the A, T, C's, and G's that you translate into amino acids. Very quickly, as virus populations evolve, some of that sort of chain of custody can be lost in the primary sequence. And so they highlight two families here. One is the ESV-1-7-like family of VFAMs, sort of alphabet soup, for sure. These are cysteine-rich proteins generally of unknown function, and they are known to be encoded or enriched in a virus that infects Ectocarpus, the brown algae, sort of the model system. And so now they can, with that sampling of 100 species of algae, they can kind of spread across that tree and then really look for those things, again, correlating to are they depleted or not found in the first place in the unicells versus are they sort of in abundance in the multicells. So that gets to a domain. They also there's a concept here of codomains. So this is sort of the mixing and matching, almost the plug and play of different sort of repeat regions with other functions that might give clues about how these protein or these genes are coming together to encode new proteins that could sort of have these new functions or have functions that sort of enhance some of the features that they're thinking about. Again, because a lot of this is like unknown functions, it's repeat families that can just be involved in interactions that are pretty promiscuous. Nailing that down to multicellularity or sort of proximal cause versus sort of an outcome of other things that are going on as these things are spreading across the world, hanging out in polar regions, hanging under rocks, despite places where the waves would normally just like crush you or just move you away from those environments. A little bit harder to say. The other family that they bring up are the FNIPs, the FNIP domains. These are leucine rich. So cysteines are an amino acid, leucines are an amino acid. These domains are enriched for those single amino acids. And these have been spotted previously in the genomes of amoeba and of giant viruses. There's some giant viruses that encode like hundreds of these genes that encode these FNIP domains that are leucine rich. Again, we're sort of just glimpsing at what functions might look like. This is where the codomain sort of analysis comes in handy. So often these FNIP domains are next to so-called FBOX domains, which are implicated in protein degradation. So this process of ubiquitination, where proteins get a protein tag that sort of targets them for removal from the cytoplasm through a bunch of protein machines. Okay, so this is great. But again, for me, at least, this raises that question. There's a lot of immunity that is done through these through protein degradation, right? So if you're a virus, you show up in the cytoplasm. If you encode proteins that can tag immune system proteins to be removed, like that's a great thing for a virus. If you're a host, if you encode a protein that can tag a virus protein to have it removed, that might be great for a host to sort of turn down the volume on virus infectivity. So at least from my kind of quick reading, it wasn't clear to me outside of that enrichment in multicellular ones, whether this is more a story about immunity versus multicellularity. So, I think some open questions here. With all of these virus gene sightings, how many are functional? I mean, this is another point they raise kind of at the end is, and they're not, to their credit, they're not making claims that everything they find here is going to be sort of like, that's the key feature. It's contributed 4% here to multicellularity, 10% here to multicellularity. A lot of this is probably just the raw material of evolution. like with all mutations, we'd expect sort of as a first approximation that most of these things as they arrive in host genomes and algal genomes in this case will be slightly deleterious or neutral. It'll be that rare one, the 1 in 100, 1 in 1,000, 1 in a million that is actually useful and gets natural selection acts on it. And that leads to an adaptation that is sort of an advance for some sort of function or ability to conduct life in an environment where there's a lot of resources or untapped resources where it's hard to live, for example. So yeah, and then how do we tie or how many of these, as we've already been, I think, harping on, how many are tied to multicellularity versus immunity versus reproduction, or some swirl of those three? So in the end, I don't know if, like, in terms of that storytelling or just sort of cutting through all of the information to meaning, how far we got. But my God, we now have this like rich, so as someone who's interested in immunity, I'm now, already daydreaming about, are there ways to mine these genomes to pull out some really cool new functions that might teach us how immune systems work? In some of these cases in ways that we haven't even seen before. Or there's some echo of some of the stuff that we see in our own immune systems, but are then now deployed in new ways. And can we I think that's really the challenge here, not just for thinking about multicellularity, but what do we do with genomes in general? How do we get meaning from them? And so are we developing tools or ways to connect that to experiments, to test not just raise hypotheses, to test them, and to even find ways of picking phenotypes. What are key outcomes or sort of results, manifestations of the content of those genes? What are they doing, and can they teach us about all this new biology that we're just glimpsing at in some of these genomes for the first time?
So you think the endogenous viral elements are bona fide?
Yeah, some. It depends, right? So, that's where I would, as we're continuing our podcast peer review, having some long read sequencing where you've got, those really good clean transitions from what is for sure kind of algal sequences versus the endogenous sequences. Endogenous sequences, that can be a little bit different from horizontal transfer. So endogenous viruses, right? So this is sort of on the edge of, we talk about retrotransposons, these selfish gene elements that are like in our genomes, that's like a quarter of the action for the classes of lines and signs, the long interspersed and the short interspersed nuclear elements, all the way to like viruses that might have been exogenous, meaning they move from cell to cell, from transmit from host to host, from individual to individual, to ones that don't actually get out of the cell. They replicate within the genome, and they're basically propagated just by reproduction, right? And that kind of blurs the horizontal transfer from the vertical in some of these cases. I think there's no question that that's happening. There's just the abundance of some of these things. But again, what's sort of genetic signal from genetic noise, which is just sort of the relics in a lot of cases of these things that might have been propagating even in some cases millions of years ago, but sort of petered out. Certainly they point this out. I mean, there's all these kind of genetic train wrecks in a sense, where things have gone off the rails in terms of the ability to sort of reproduce. And so they just sort of hitchhike along with the rest of the genome and sort of are just sort of detritus in a sense. And that's an actively debated field right now, is how much of that is like sort of junk DNA versus, has real functions. So
this reminds me of a story that Matthias Fischer told me when I was in Germany in December. I spoke with him. He said, these virophages, right, that are associated with giant viruses, they have to co-infect the same cell. He found one that had been integrated into the host genome, right? And so I asked him, are there others? He said, well, that's a good question, because for a long time, we had all these genome sequences of hosts, but we didn't see any virophages. But the problem is that the viral sequences were thrown away by the algorithm.
Exactly.
And only when you went back into the raw read archive did you see that they were attached to the genome, because the algorithm was trained, if you get repetitive elements, which some of these are, you just throw them away. And so, you can get all this sequence data, but you have to analyze it properly and often in unique ways that are not done by off-the-shelf programs, right?
Yes, and that's a super important point here, because I think it's, we're in this moment in science too, where there's all of this excitement or hype around artificial intelligence. But as you train those off-the-shelf models, will they detect new patterns, some of which are like totally new, potentially even breakthrough discoveries? Absolutely. But in some sense, they're only as good as the humans to begin with, the humans that have trained them. And so, putting human beings back into this process as much as possible, that to me feels like a real opportunity where, if you really care about one of these 100 species, because you just can't get it, like you've spent a career thinking about the unique and, beautiful biology from sort of one of these exotic algae, or it conducts some function that's like a great model for something else, then you're sort of incentivized to, like, you see that genome and it's like a gold mine, right? You're motivated or incentivized to get to that point of meaning. And so I think that's where putting the humans back into, and certainly the authors are humans doing this as well. They're kind of, I think, kind of building a scaffold in a sense to sort of think about, here are some, rough contours about what's enriched or what's here, what's correlated with that. But I think where the real, sort of breakthroughs might emerge are when we move beyond those sort of first impressions into really digging in, and sort of seeing something and saying, well, this is worth chasing and figuring out. That's where the human intuition really can, I think, move some of this analysis in great ways to the next level. It's also, this is important too, because we're in this moment, maybe it's starting to fade a little bit, but where there's real motivation to just like use these tools. Like if you have the old cliche, if you're a hammer, everything looks like a nail.
Yeah.
You just want to sequence everything. And then the danger here can be if you sort of plant a flag and say, we just sequenced these 100, maybe not algae, but let's just say like we sequenced this new, the genome from a primate that had never been analyzed before. And but you don't add much value to it, like the annotation might just be a kind of off the shelf, out of the box. Then the danger here is you've kind of planted a flag and said, well, this genome is done. And then imagine someone again who like really cares about that species, because it's like the perfect model for thinking about this sort of, specialized biology or something like that. That person might go to a granting agency and be like, we really need to invest in this, like the genomic tools here. But someone on the other side of that, a peer review, might say, oh no, that genome was done 3 years ago. When we say a genome is done, there's a real like possible like landmine that emerges there. And so that's where, these conversations are important to really understand the difference between sequencing something and understanding it. And there's a lot of ground between those two things. And where I think we need to be careful as scientists, as peer reviewers, et cetera, is to really try to carve the space for that single lab, or those, that human who just loves that species or that biology that they see in that species, to be able to have the resources to continue to build that in this genomic space. That's
I think, data like this need to be published so people can look at it, right?
Yep.
Even though, as you say, there's not a compelling story, perhaps, still it needs to be out there so that other eyes can look at it and think about it, right? That's so important in science.
Absolutely. And don't get me wrong, I think there is a story here, but is it how it's sort of, to me, it feels really like a kind of flying over the top story versus really digging in and interesting. And so there's just different stories out there. None, and I'm not trying to say one is better than the other. I think they're valuable in different ways, but that's really the opportunity here to unlock, with all this information, to unlock something new. And are we able to do that going forward?
We have a couple of comments here.
Great.
So let's see. Abdur Rahman says, the whiteboard looks exactly as I would imagine for a macroalgal discussion. There you go.
Yeah, the conspiracy theory, all the lines and confusion and chaos are playing out in many forms and functions.
So Don says, is it more expensive to do longer sequencing, or are there other reasons not to do it?
Yeah, this is great. So, I think expense is less of an issue now. Like that's really one of the great things about the rapid advances of this technology, is it's really like in reach. It's more about the pipelines that exist. And so the short reads have been around longer. And so, the computational tools that have been developed around that are sort of tuned to that exact technology. What's emerged, but it's lagging, it's catching up, and in some cases really fast, is the ability to sort of do the same level of analysis on long reads, or better yet, to merge these sort of data pipelines, the short reads and the long reads, and to really sort of get the advantages of both, and to sort of cover the the sort of shortcomings of both types of technology there. That is starting to exist too, but it's still relatively in its early days. And so, especially at this, like if you're sequencing 100 things, there's some real reasons to go kind of big with your limited resources, to go big into the short reads. I don't sort of begrudge that of the authors at all. But then, as you're maybe doing more kind of, between 1 to 10 genome sort of level of analysis, that's where I think the long reads can really add a lot to the conversation. And that's where, I think we're also in this important moment where computational biologists, experimental biologists, or, kind of everything in between, to sort of team up in ways that are kind of, are intellectually engaging for both sides of the equation. That's another thing that is sort of an ongoing opportunity and challenge that I think we're wading through in the field of modern genetics and genomics.
So John says maybe with longer reads it would be harder to compute out retroviruses.
Yeah, so I think, so that's possibly the advantage of the long reads, is like, let's say there's going to be kind of everything you could imagine, entire retrovirus genomes integrated into the host genomes, right? Portions of them, free retroviruses, or viruses that encode I'm gesticulating too much. There's this sort of bug in Apple right now where it gives you that thumbs up. That's how I know I'm like awake and engaged here, is when I involuntarily put a thought bubble with a thumbs up there. Actually, long reads can be really useful to sort of get at what the sort of various forms of the viral sequences are. But also, I mean, there are some disadvantages of long reads too, right? So in a lot of cases, the error rates are a lot higher. And so getting high quality genomes can be challenging. And so that's where kind of merging these lanes is sort of the best of both worlds currently. Great question.
Let's see here. Pete says, TWiEVO number 101 discussed loss of tails in the higher apes. This topic was taken up in the local BBC Science podcast too. You're in good company.
Awesome. I love it. Yeah. It's a pretty, that's what we're trying to do on TWiEVO. It's kind of like a tasting menu where we're jumping in between. Here we're thinking about kelp, whereas last time we were thinking about the tails, or the remnants of our tails, and what that means in primate evolution. We're kind of that's the hope, right, Vincent? We're all over the map.
Yeah.
And by the way, learning as we're going too. That's kind of what keeps us in the game, is all of this, like this wealth of papers on all topics. And so, yeah, we're probably converging or hitting on some provocative themes here.
Andrew says there's also the problem of getting rid of waste products if you're not at the surface of an organism, right?
Totally agree. Yeah, as you go farther down that road of multicellularity, and that's kind of that interesting possibility, I think, between the brown algae that they point out that only kind of came on to multicellularity in the recent past, versus if you've been doing this for 1 billion years, those problems of waste products, or of getting nutrients and disposing of waste, like you're probably farther along that pathway in a sense if you've been multicellular for longer. And so there's some interesting possibilities to kind of compare and contrast the sort of sophistication of multicellularity in some of those cases.
So Don asks, do long sequences of leucines tend to fold in some particular way?
Yeah, so that's like same with the anchor and repeats. So a lot of times it's almost these proteins become quote unquote sticky somehow with each other. Getting specificity out of this takes a little bit more than just sort of finding these things. This is where I think some of the we're still kind of in the infancy in a sense, but some of the computational predictions of protein structures, of how these things fold up, can be really informative. It can also be sort of frustrating where you get all these quote unquote what are called disordered regions of protein. So we think about proteins so we go from that primary amino acid sequence, which is just the 20 letters of the 20 letters or so, the amino acids that we know of, or that are commonly used in proteins, to fold into that 3D structure. And so as we do some of these predictions or even some of the structures, there's like, these are not just globular proteins they're breathable. These things are sort of folding and unfolding in some cases in real time. And so so-called disordered regions, a lot of the computational predictions just sort of punt on it, and you get these like long kind of spaghetti sort of representations that probably don't really represent the folding and unfolding here. And so, yeah, it's sort of, these surfaces that are kind of enriched for the ability to interact with other proteins or to breathe, to move in real time, and sort of conduct this kind of all of these layers of biological organization. One of them is at the protein level, and we take all the, like, I think historically the last 15 years we've got all these tools, structures, X-ray crystal structures, all the way to electron cryo-EM, or like you basically freeze these proteins and then try to look at them and use computers to decode what they look like at high resolution. These are all snapshots. And so the ability to get closer to real life, which is the movies that these proteins are playing out right in real time, that's still in its infancy, I would say.
So ZZ Captain says, should we trust viruses? They seem tyrannous.
This is great. Yeah. No, we shouldn't trust viruses. What have we just lived through for the last 4 years? I don't trust a virus, but I have a lot of reverence for viruses. And as a geneticist, how they're, really just kind of poking biology along, provoking biology by providing all of this genetic diversification. Without, I can guarantee this, without viruses, we wouldn't be here. Sort of the,
not just not podcasting, but existing, right?
Yeah, that's right. And so that's really, I think, one of the fun, both kind of this crosses between sort of the philosophical and the scientific, is as we think about like, who are we? If we think about our genomes, how much of that is seeded to viruses, virus-like particles, endogenous viruses, or retrotransposons, that far outweighs the sort of protein coding genes that we would think about as like being us. Viruses have really written the book here and have, if anything, outnumber us. So it may be an analogy to draw here as microbiome, right? Where by weight, as we're sort of sitting here podcasting, we're more microbe than we are our own cells, just given all of if you sort of dissolve away all of our cells and just leave the microbial ones, you'd still see an outline of us. And by sheer weight, that sort of out. And so it's a different way of thinking about what it means to be a life form. It's the conglomerations and the collaborations, sometimes positive for one actor and not so positive for the other, that are sort of underlying all of the complexity of life around us.
Kang wants to know, are the same junk DNA sequences found in other species?
Absolutely. And in some cases off the charts. So like, I think if we look at some of the lines and signs of our genome, it's like 25%. If you look at plants, corn, maize, Barbara McClintock, is sort of the pioneer here. 3/4 of the genome might be, sort of junked up, so to speak, with these transposable elements, the jumping genes that had. And so there, the magic of McClintock was linking that genetics, the genome composition, to the phenotype. And the phenotypes were beautiful, right? These are these corn cobs with these different color kernels, or the sectors, the different pigment that sort of comes out as a result of those transposable elements. What McClintock had was both kind of that genetic intuition, like decades in some cases ahead of her time, but also had the phenotype, these beautiful outcomes, those, sort of really cool patterns that you could sort of see the consequences in real time. That's the trick here, right? Is what's the phenotype? Multicellularity, obvious phenotype, but how do you link that sort of directly to each genetic element? That's a lot more challenging in a lot of cases.
What's the phenotype? Reminds me of that famous line, what's the frequency, Kenneth?
There you go. REM, I think. Good music
all right.
reference there. Yeah.
Want to do some picks, Nels?
Yeah. Let's do some science picks of the week. So I've got one, Vincent, so as we're thinking about the impact of viruses and sort of that blur between art and science, or history and science, I just saw in the Washington Post yesterday article, a short piece in their science section titled 12-Sided Roman Relic baffles archaeologists, spawns countless theories. So here's the artifact or the relic that the archaeologists dug up. I think this is somewhere near the UK. What do you see there, Vincent?
It's a virus, Nels, of course.
Yeah, dodecahedron. This looks like an adenovirus to me.
Could be, well, any icosahedra fall under the dodecahedra, right? Classification. So any of these icosahedrally constructed viruses, you see the pentamer there right in front.
Yeah.
And they have little blebs at the fivefold axes. Actually, that's not the fivefold. Actually, it looks like the threefold axes. But
yeah, that's right.
What are the speculations about what it might be? What are they saying?
It's all over the map. So this is a bronze piece. And so all the way from kind of wild ideas about, and I think like echoing sort of the functions of virus structures, it was just carrying things. Would people put seeds in there and then somehow, plant the fields somehow, like using the thin evidence of that? Was it, was it used as in a game or something like that? Or is it just for the artistic part of it?
Could be.
Yeah. I don't think anyone knows.
I got something. Yeah, let me get this from my shelf.
Yeah.
So I bought this. I bought this at like West Elm, the furniture.
Yeah.
company.
Yeah.
This is an icosahedron, right? And it was labeled in the store decorative element. So that's what I bet that's what they had. They had a decorative element. They had discovered geometric solids. And so it's the same thing. I wonder how much it cost.
Exactly. I mean, to me, it's just like I saw that image. It's just it's artistic, right? It's just
it's beautiful,
aesthetically pleasing.
Also, the holes are different sizes. You notice?
Yeah. Interesting. Yep.
Andrew says maybe an incense burner.
Yeah.
Holes, right?
Yep. I like that idea. But I love that how it also just so nature has sort of hit on like what our brains perceive as aesthetically pleasing or useful potentially for, various uses that I think people are just wildly speculating at. An echo of biological organization that the viruses have hit on that, of course, this is 1,000+ years before we actually saw some of the eerily similar structures of virus particles. But it kind of ties this all together somehow.
So Jan says it's an early disco ball.
There we go. Yeah. Now we're talking.
Pete says there's strange stone geometric balls from the Neolithic era, some thousands of years older.
Exactly.
Yeah, humans do interesting things. And Hey MJ says sometimes the simple answer is the right answer. It could be. So what's the simplest answer? It's a decorative element, right?
Yeah, just look exactly.
So an artist made it, and they thought it looked cool. And as Don says, Don sees a regular polytope, all of which were known to the ancient Greeks. So this is known, and artists can just make it. Why not? Yeah.
Yeah. And then the viruses picked up on it somehow too. Some of these forms have functioned pretty effectively for propagation of genetic material.
Yes, it is a main platonic form of the platonic solids. That's right.
That's right.
Absolutely.
So that's my pick of the week. And I think we'll have a link. If you don't want to sort of chase after that article, we'll have a link to it in our show notes. Yeah, pretty fun.
Yeah, Rich Condit sent it to me, and he said, they're puzzling over what it is, but I know what it is.
Yeah, there you go. Our TWiEVO scouts are on the case. Kathy and Rich on this episode. How about you, Vincent? What's your science pick of the week? So
I want to highlight Nicole King's work. So we interviewed Nicole back in September 2016. Yep. And here we have her episode, TWiEVO 11, Microbial Accomplices in Multicellularity. So as Nels mentioned, she works on choanoflagellates, which are the closest living relatives of animals. And this is an interesting organism because it can become multicellular and in nature and in the laboratory. And she studies that. So it exists as single cells or as a multicellular organism. As you can see, they are a colony.
Yeah.
And she has found that this development is regulated by bacterial lipids.
Yeah.
So that's very cool. And then I looked to see what she's done with that. And there's there's a more recent paper, which I will make my co-picks. This is a
Cool.
applied environmental microbiology paper, synergistic cues from diverse bacteria enhance multicellular development in a choanoflagellate. So the thing is that, let's see if there's some nice pictures. Yeah, here you go. So you can see they can exist as single cells here in panel A, and then in panels B and C, you see they're aggregating into, I guess, simple multicellular organisms, right?
Exactly.
And the finding here is that lipids from many bacteria can do this, which makes sense, right? Because it wouldn't be one just in nature, right?
Exactly. And I think an important, breakthrough level concept here, which is as we think about our own genomes and sort of trying to distill it down. So here we have the concept of horizontal transfer, all the contributions of viruses, et cetera. What Nicole's work and others are saying is don't totally distill it down to that one genome. It's in those interactions that some of these transitions, and especially when we think about the evolution of those transitions, that's sort of central to the story here. This also, Vincent, great pick because I think Nicole was just inducted into the National Academy of Sciences. And so, we're good evolutionary talent scouts, right? We had Nicole on, I think something like almost 8 years ago, and her career continues to skyrocket for great reasons.
Talent scouts. So also inducted was Marion Koopmans.
Yeah, I saw that.
Stan Lemon and Paul B. Nash, all of whom have previously been on TWiEVO. So what's the moral? That we get them first.
That's right.
We recognize their talents first.
Yeah. Try to get that invitation to this week in podcasting because only the sky's the limit. That does remind me, Vincent, it's been a minute since we've had a guest on the show. I think it's probably time for us to see if we can get a third voice to join us and to share their science. It's always, totally fun to hear the backstories and also hear it from the voice of the scientist who actually knows what's going on.
We had a guest in 100, remember?
Yeah, that's right. Exactly.
That was a lot of fun. And she showed us the museum. That was so cool.
Yeah, that was great. And I have a report there. So the glove box, it was sort of in mid construction. It's advanced. Maybe I'll try to get a photo on this. Maybe this will be my pick next week to describe its development. It's really come together in a cool way. It's just even in the last few weeks.
Yeah, it was a lot of fun. And, it was technically challenging to do that, but we pulled it off. It was great.
It was fantastic. Really, agreed.
All right. Yeah, as Tom says, for authoritative sources, Microbe TV.
I'll drink to that.
We got a bunch of scientists here who are all looking for cool stuff. I mean, how can you go wrong, right? These scientists that I work with here, they know their stuff. So,
and are learning as they go too, and that's
always.
part of the conversation, which is so exciting.
All right, that's it for this episode. You can find the show notes at microbe.tv/TWiEVO. If you enjoy our work, we would love to have your financial support. You go to microbe.tv/contribute. Also, you can go to Venmo, which you can see the name up there on the top, or you can hit the donate button right under the chat screen. Anything you want to do, we need your support to make more science programs. And we have some interesting ones coming up, including a new Science of Exercise podcast with Angela Mingarelli and her co-hosts. So look for that in the next couple of months.
Very cool.
If you have any questions and comments, TWiEVO at microbe.tv. Nels Aldi's at cellvolution.org. L Earlybird on Twitter. Thanks, Nels.
Thank you, Vincent. Great to be together, and thank you, everyone, both financially and intellectually, for contributing to the conversation here. Let's keep it running.
I want to thank our moderators for today: Barb Mack, UK; Andrew from New Zealand; Tom Steinberg from Oregon. We had Vanity Nutrition, and I believe that's it for the moderators. Thank you so much. And thanks. And Les, of course, was here for a while.
Barb Mack, UK, was here for a bit.
Yep. Thanks for joining us, everyone. Tonight, 8 p.m. Eastern, which is in like 5.5 hours from now, we'll have office hours with just me. But, we have fun together. So if you're staying up, check that out. That would be fun. Let's see. I thanked everybody. I thanked the moderators. I thank Nels. Now it's time to turn on the ending music because you have been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious. Bye-bye.